The overall goal of this procedure is to establish a chronic Salmonella-infected mouse model. This is accomplished by first preparing a Salmonella solution. The second step of the procedure is to gavage mice with streptomycin, followed by Salmonella or with streptomycin only. The third step of the procedure is to isolate Salmonella from mouse fecal matter and detect Salmonella infection in mouse intestine using BBL chromager plates. The final step of the procedure is to observe and record mouse body mass and note changes in vitality following Salmonella infection. Ultimately, results can be obtained that show an infection of Salmonella can be established in the intestine of mice gavaged with Salmonella.
Hi, I'm Shaoping from the laboratory of Dr. Zhang in Department of Medicine at University of Rochester.
I'm Rong, also from the same lab.
And I'm Yong-guo Zhang from the same lab.
Today, we will show you a procedure for establishing a chronic Salmonella-infected mouse model.
We use this procedure in our laboratory to study host bacterial interaction, signal transduction, and tumor genesis.
So let's get started.
Before experimentation, prepare enough Luria bertani, or LB, broth plates for each Salmonella strain and to use throughout the experiment. The plates can be refrigerated and stored for several months. Next, prepare Salmonella culture. Remove a vial of stock Salmonella, frozen at -80 degrees Celsius, and let it thaw on the countertop. To plate Salmonella, flame an inoculating loop in a Bunsen burner and dip into the thawed bacterial stock. Zigzag the loop back and forth on the top third of the LB plate. Flame the loop again and